Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall look at covalent modification as a way to regulate enzymes and therefore a way to regulate metabolism. In the previous videos, we saw how we can regulate metabolism by sequestering enzymes within certain compartments, by the availability of cofactors, as well as by allosteric regulation. Recall from the previous video on allosteric regulation that the chemical that will regulate the enzyme, what we call the modulator, which could be an activator or an inhibitor, this chemical will bind to the enzyme at the allosteric site, and this binding is non-covalent interaction. However, in covalent modification, the enzyme, the chemical that will bind to the enzyme, in this case, for example, the acetyl part, this will bind to the enzyme covalently. So this is covalent modification. Besides the addition of the acetyl group to the enzyme, what we call acetylation, enzymes can also be methylated, methyl groups added, hydroxyl groups could be added, that's called hydroxylation, and AMP groups can be added to the enzyme, that's called adenylation, as well as several other groups can be added to the enzyme. As with allosteric regulation, the covalently modified enzyme may show increased enzymatic activity or it may show decreased enzymatic activity. The most common covalent modification, and the one we will focus in this video, is phosphorylation, where the hydroxyl group present in the enzyme, and remember enzymes are proteins, so hydroxyl groups could be found in threonine, serine, or tyrosine amino acid side chains. And these hydroxyl groups are covalently modified by attaching one or more phosphates. This is called phosphorylation. This reaction between the enzyme, the protein or the enzyme, and ATP to give us ADP and the phosphorylated protein, this reaction is carried out by enzymes that we call protein kinases. There are several different types of protein kinases that we'll come across in future videos. If the tyrosine was phosphorylated, we could call the protein kinase as a tyrosine kinase. If the serine was phosphorylated, it would be a serine kinase. If a threonine was phosphorylated, then we could call it a threonine kinase. The phosphorylated enzyme could either show increased catalytic activity compared to the non-phosphorylated form, or it could show decreased catalytic activity. Now, this phosphorylated protein can also become unphosphorylated. So the phosphate groups can actually be removed to give us back the non-phosphorylated protein. And this reaction is carried out by enzymes called protein phosphatases, which help remove the phosphate groups from the phosphorylated protein to give you back the non-phosphorylated protein. Now, it's not surprising that this is the most common and the most important covalent modification is the most important way to regulate enzymes, because this is connected to energy, ATP. So as we mentioned, emphasized many times in previous videos, ATP, the cellular energy present, is very important in regulating metabolism, as well as many other biological processes that we see in our cells. The basic concept of phosphorylation is relatively straightforward. So for example, here we have a non-phosphorylated enzyme. Suppose now we added a phosphate to this hydroxyl group. Now there's this phosphate. Remember, phosphate gives us negative charges. So this area will be negatively charged. And let's say there was some positive charges nearby. So this is going to result in an attraction between the negative and the positive, an electrostatic attraction. And that's going to change the shape of the enzyme. And as the enzyme shape changes, the activity of this enzyme may either go up or it may go down. Further phosphorylation, for example, this group becomes phosphorylated. Now we have negative charge here. And let's say there was another negative charge nearby that would lead to repulsion. That will cause a further change in the enzyme shape, and that would lead to a further increase in the enzyme activity or a further decrease in the enzyme activity. So let's apply this concept of covalent modification using an example. We use two enzymes from glycogen metabolism. Remember, glycogen metabolism, as we discussed in previous videos, is very important, especially in the liver as well as muscles. We'll look at glycogen synthase, that will abbreviate GS, and then we'll look at glycogen phosphorylase, that will abbreviate PYGL. So recall that glycogen synthase is the key enzyme that is involved in glycogenesis, which is glycogen synthesis, while glycogen phosphorylase is the key enzyme that's involved in glycogenolysis, the breakdown of glycogen. So here are the two enzymes, glycogen synthase on the top here, glycogen phosphorylase, which is involved in glycogen breakdown at the bottom here, shown side by side so that we can compare the two enzymes and what happens to them when they are phosphorylated. Glycogen synthase is phosphorylated by a variety of protein kinases, the two most important being CAMP-dependent kinase, which is protein kinase A, as well as glycogen synthase kinase 3, or GSK3. There are other proteins that can phosphorylate glycogen synthase, but these are the two most important that we know of today. Now, when glycogen synthase is phosphorylated, where the hydroxyl groups become phosphorylated, the active form of the enzyme, which is the non-phosphorylated form, is converted to the inactive form of the enzyme. So since these kinases make the active form to the inactive form, we can consider these kinases to be sort of inactivators of glycogen synthase. Now, the inactive form of glycogen synthase can be converted to the active form by removing the phosphate groups. And this is done by the enzyme called phosphoprotein phosphatase 1, which we shall abbreviate as PP1. Since PP1 converts the inactive to active form of glycogen synthase, we can consider PP1 to be an activator of glycogen synthase. Now, in case of glycogen phosphorylase, the inactive form is the unphosphorylated form. This form can be converted into the active form by adding phosphate groups. And this is carried out by the enzyme called glycogen phosphorylase kinase, or PHK. So PHK basically converts the inactive form into the active phosphorylated form. So it's just the opposite of what we see above. For glycogen synthase, the active form was the unphosphorylated form. For glycogen phosphorylase, the active form is the phosphorylated form. So since glycogen phosphorylase kinase, or PHK, makes the inactive form into the active form, we can consider PHK to be an activator of glycogen phosphorylase. We sometimes refer to the active form of the enzyme as the A form and the inactive form as the B form. So in this case, the active form here is the A form and the inactive form would be the B form. Now the active form of glycogen phosphorylase can be converted into the inactive form by removing this phosphate, and this is done by the same protein that we discussed here, PP1. So since PP1 makes the active form into the inactive form, we can consider PP1 to be an inactivator of glycogen phosphorylase. So to summarize, glycogen synthase is inactivated by pKa and GSK3, while it is activated by PP1. On the other hand, glycogen phosphorylase, which is involved in glycogen breakdown, is activated by PHK and inactivated by PP1. So PP1 inactivates glycogen phosphorylase, but PP1 activates glycogen synthase. Now, covalent modification is often seen in cell signaling, where hormones as well as other signals, when they bind their receptors, they lead to this process called cell signaling, or the signal transduction pathway. And we'll talk about this in future videos. But for now, let's just use one example. We'll use insulin as a signal. It's a hormone, and we'll see how insulin regulates glycogen metabolism by regulating this enzyme glycogen synthase, which is involved in glycogen synthesis, as well as by regulating glycogen phosphorylase, which is involved in the breakdown of glycogen. So recall from previous videos that the insulin levels in the blood only increases when your blood glucose is very high, which means after a meal. That's the only time when the blood glucose becomes very high. So at that time, insulin is secreted by the beta cells. Insulin levels are high in the cell. So there is a better chance for the insulin now, because of its higher concentration, to bind insulin receptors that are present on cells. And when the insulin binds insulin receptors, that will trigger the signal transduction pathway, cell signaling, and this cell signaling will lead to increase in glycogen synthesis, increased glycogenesis, while at the same time decreasing glycogen breakdown, decreasing glycogenolysis. Now, glycogen synthesis increases. How does that happen? That happens by increasing the activity of glycogen synthase. So this enzyme activity goes up, increases when insulin binds to its receptor. So what makes the glycogen synthase activity go up? Well, there are three major ways. One is inhibiting the inactivator for glycogen synthase, which is GSK3. So this is an inactivator of GS. So by inhibiting this, which is the first process here, by inhibiting this inhibitor, this enzyme activity is going to increase and glycogen synthesis will therefore increase. Another mechanism is by inhibiting another GS inactivator, and that is pKa. So pKa is an inhibitor of glycogen synthase. So if we could inhibit pKa, and that's what happens when insulin binds, again the glycogen synthase activity is going to increase. A third mechanism is to stimulate glycogen synthase activator PP1. Now, PP1 is an activator of glycogen synthase. So if we could activate PP1, and that's what happens when insulin binds, it activates PP1. and PP1 will activate glycogen synthase, and glycogen synthase activity will now increase. So through these effects, the binding of insulin to the receptor is going to lead to increased glycogen synthesis. So while glycogen synthesis increases, this enzyme activity increases, insulin will also at the same time decrease glycogen breakdown. It will decrease the activity of glycogen phosphorylase. So how does it decrease the activity of glycogen phosphorylase? There are two major mechanisms. One is by stimulating the inactivator of glycogen phosphorylase, which is PP1. So by stimulating PP1, and PP1 is an inactivator of glycogen phosphorylase, that is going to cause a decrease in the activity of glycogen phosphorylase, and therefore glycogen breakdown slows down. And another mechanism would be to inhibit the activator for glycogen phosphorylase, and this activator is PHK, glycogen phosphorylase kinase. This is an activator for glycogen phosphorylase. So if we inhibit this PHK, and that's done here through PP1, that will lead to reduced activation of PYGL, and that will lead to decreased activity of glycogen phosphorylase. So this enzyme activity goes down, this enzyme activity goes up. So in this way, hormones such as insulin can increase glycogen synthesis in different cell types and at the same time decrease glycogen breakdown. We shall see more examples of this as we discuss regulation of metabolism after a meal and regulation of metabolism during starvation periods in future videos. Now in the next video, we shall look at another way of regulating enzymes, and that is by having the enzyme in an inactive form that we call zymogens. So until the next video, once again, thank you very much for watching and supporting this channel. Have a wonderful, wonderful day, and we'll see you soon in the next video.